I'm Raghavendran. I'm working as a bioinformatics mentor with Pine Biotech, and I welcome all of you to this portion of the webinar to introduce you to this hackathon. And let's discuss some basics about in this portion. We will discuss some basics about what is microbiome and what are some of the sequencing technologies that relate omics data analysis with microbiome, and what is the approach in bioinformatics analysis methods towards understanding the association of microbial population with disease. And we discuss this from the point of view or from the popular publications and popular research reviews. So this will actually give you necessary tools and some idea and introduction, and this will prepare you for participating in this hackathon that focuses on microbiome in health and wellness. So let's jump in. Let's start by reviewing a little bit of history of microbiology. So where the study of microorganisms leads to the creation of tools and approaches that are still used today to continue and to uncover the veil of world which we do not see, but yet has such a profound effect on everything that is around us. This all started with, for example, microscopes that helps us observe that actually helped us observe the first bacterium in 1673 itself. But today we have microscopes that can enlarge from 50 to 300 times the size of the first observation of bacteria that was made long time ago. So as the resolution of these microscopes improved, more and more representatives of this unseen world have been discovered. Today, the microscopes that we are using are tremendously powerful, and it allows us to see smaller and smaller elements that reveal how the world of microorganisms functions on every imaginable surface. So in this example, for example, in this image, we can see a super high resolution image of bacteriophages that are green in color attacking the bacteria, that is E. coli, that is blue in color. So we have advanced to such a level of high resolution, and this actually made us define microbiology not only by the organisms that the research studies or that the field studies, but also by the tools that are used to study them. And these tools, as we already discussed, has evolved to allow us to study and categorize microorganisms specifically. And the technology for bacterial culturing, coupled with the technique to stain bacteria, has historically improved our ability to isolate, to classify, and to determine whether a bacteria is a cause of human disease, or even select appropriate treatment for handling that pathogenic entity. Why are we interested in studying the microorganisms generally? So the connection between disease and microorganisms has been well established since the 1800s. And these studies actually provide important information about bacterial function in isolation. However, as more and more studies came through, it became apparent that most microorganisms live in a complex communities, and many members of which are impossible to culture in isolation. And today, we know that microorganisms, or the microbiota that make up the, for example, human microbiome, are not just unicellular organisms living in isolation alongside each other, but instead they are highly regulated, highly structured, and highly functionally organized communities. So let's take human skin as an example, right? Bacteria are, we know that bacteria are able to grow adhere to almost every surface, forming architecturally complex communities that we call, or that are termed as biofilms. The stability of these biofilms or the communities is fascinating, as many of us probably know from trying to battle common skin conditions such as dry skin, acne, and eczema, for example. So forming these distinct communities, these microorganisms are interconnected and are determined to maintain their community stability. And these bacteria, for example, within the community or from this biofilm, can communicate with each other by producing, detecting, and responding to small signal molecules in a process called quorum sensing, right? This process, or this quorum sensing activities in these communities, are also involved not only in communication, but also in the virulence of the pathogenic bacteria, or in response to, for example, external stimuli, or also in controlling the host defenses, and in responding to antimicrobial agent, etc. So inside such community, or inside each community, there is a balance of bacteria, fungi, and viruses that controls the species abundance and the activity. So this balance is very absolutely necessary and very important for our understanding of the association of this microbial community with not only with the diseases, but also with health and wellness and general well-being of ourselves. So microbes inhabit just about every part of the human body, not only on the skin, right? For example, we already saw some examples of microbes living on the skin, and we definitely see microbes living in the gut and up in the nose and gastroenteric pathway everywhere. The microbes, or the diversity of these organisms that inhabit various human bodies, and are estimated to make up more than half of people's cells, right? And this diversity is extremely critical to human health, right? And most of the time, these microorganisms that live in harmony with with their human host provides extremely important functions that are essential for human survival. For example, from enabling the work of the immune system to underpinning brain function, the microbiome has been linked to nearly every health condition that one can find the association. So today, health-related interventions and well-being related interventions seek to manipulate the population of this microbial diversity, or the diversity of this microbial population, rather, through products such as probiotics, through drugs, through therapies, through foods, and through wellness products, and etc., etc. Some of which Ilya already had introduced you. So one such example in this association is detailed in this review, right? This review focuses on the microbial diversity in the gut, altering the responses in the diversity observed in the skin, right? For example, this image actually shows the inflammatory and microbial influences between the gut and skin for a healthy state in the left, and for the dysbiotic state or non-healthy state in the right. And they are compared in detail in this review. So I will shortly summarize it. The dysbiotic state is characterized generally by an impaired gut barrier, for example. This impaired gut barrier results in the imbalance in gut microbiome. And this gut microbiome can be, for example, can also result in reduced mucus layer formation, or reduced immunoglobulin secretion, or this again can promote barrier disruption. This can actually create imbalance in the intestinal permeation into the bloodstream, and also can generally cause gut inflammation. Or this dysbiotic state can also be characterized by impaired skin barrier, right? This generally results in imbalance in the skin microbiome, and this reduces, for example, human and antimicrobial peptide production. This promotes skin rashes, this promotes lesions, skin inflammation, and etc. other conditions. This dysbiosis between the gut and skin are actually connected through a balance between, or balance in the helper T cells or T lymphocytes. And in this dysbiosis, or in this imbalance, this balance is actually skewed towards, this balance is actually extremely skewed, where we can or this review actually focuses on identifying whether this skewness results from the gut dysbiosis or from the skin dysbiosis. So if you want to check, if you want to read a little bit more about this review, let me paste the publications link, if I can get it, in the chat box. Yeah, I got it. So please go through this publication if you are interested in. This is an excellent review that will open your eyes to several different associations, and that can be very, very helpful in general. So moving on, the imbalance, as we saw in one such example, for example, gut microbial diversity, is related to many modulations or many infectious diseases and and their pathogenesis. For example, many more studies actually uses the Human Microbiome Project data for the following association, right? The role of gut microbiome has been associated to Crohn's diseases. It is generally found associated with ulcer or esophageal cancer, right? The role of skin microbiome is associated or is generally related to psoriasis, atopic dermatitis, and even sometimes immunodeficiencies. And sometimes in some of the publications, the role of urogenital microbiome is related to reproductive complications, right? And Elia also focused on the childbirth, right? And several publications and research focuses on understanding number of childhood disorders from gut microbial population, right? Including, for example, pediatric abdominal pain. It also includes intestinal inflammations, and also sometimes a severe condition in which premature infants actually die due to the fact that the intestine actually dies inside them. So these are all some of the interesting starting points that you can explore. So what happens is these researchers and these studies actually fuel this exponential increase in the microbial data or microbiome data that are available in publicly available resources or repositories. And these were significantly advanced through major initiatives in, for example, in the metagenomics of human intestinal tract, or in subsequent Human Microbiome Project. Now we have Earth Microbiome Project, and a lot of these huge studies that have been made possible through advancements in metagenomic sequencing, right? So the result of this is that not only skin and gut, which are well known to be a carrier of diverse microbial population, lungs and even brain microbiota are starting to be characterized. And each new project represents extremely vast opportunities and challenges to determine which microorganisms play a key role in the development of certain environmental conditions necessary for healthy functioning of the human organs, or which subset of those microorganisms can be harmful to those human organisms themselves, right? So this actually, as we know, can contribute to critical, this actually can expand our understanding of studying these endogenous human microbial communities that can contribute to critical metabolic and physiological and immunological functions that include maturation of the immune system, that includes, for example, energy generation, metabolic regulation, fat storage, and skin maintenance, and etc., right? You can explore a lot of these things in this program, in this hackathon. So far we have been discussing about the data or microbial data, metagenomics data, microbial population. So what's in store for the bioinformatician, right? For the bioinformatician's perspective, from the bioinformatician's perspective, metagenomics is the genomic analysis of microbial DNA from environmental communities, right? The tools that will enable us to perform this metagenomics analysis will actually help us understand the population of this microbial community that were previously unculturable or that were previously unexplorable, right? And this ability to identify microbes without an a priori knowledge of what the sample contains is opening new doors in disciplines not only related to, for example, disease biology, but also generally expands the horizons of microbiology, virology, environmental sciences, biomedical research, as such, right? And sequencing-based examinations of metagenomes has actually become a powerful tool for generating novel hypotheses based on the microbial population and microbial community population analysis. Oh, my animation is running slow. Sorry about that. Yeah. So unlike other omics, metagenomics does not study a single organism, right, but a whole community at once. This is a snapshot of collection of organisms directly in their natural environment, right? Since the genomes are so diverse, this is typically accomplished or carried out by sequencing a specific marker portion of the genome instead of all the genomic material present in a given sample. And how are these done? Many of these processes are done using next generation sequencing that focuses either on sequencing, sometimes sequencing the whole genome or transcriptome of the microbial population, or most of the time, a popular approach is to select a portion or select a gene of these bacteria or the portions of the genes that are in the variable sections, right? We will discuss this in a little bit while. Yeah. So these can be shortly divided or roughly divided into categorized into shotgun metagenomics or metatranscriptomics approaches and 16S amplicon based approaches, right? The 16S amplicon sequencing is actually a recently developed, it's not now recent, it's very well developed now, technique that is widely used to study microbiome directly in its environment, as we already discussed, right? This approach or this sequencing approach focuses not on the whole or not on sequencing the whole genome of the organism or of the bacterial community, but it focuses on 16S or 18S ribosomal rRNA that can be amplified using several amplification methods. This gene, 16S gene, contains hypervariable regions that can provide species-specific signature that are useful for bacterial taxonomical identification. And this identification can be carried out from a diverse set of these species-specific signature sequences. To account for the diversity of such sequences, they are usually combined into operational taxonomic units or OTUs, which can be used to classify groups of closely related individuals. And OTUs are actually formed from groups of these genomic sequences that represent a cluster or a phylogenetic clade that has that are closely related and that has meaningful abundance in our sample. So by far, the most common type of clustering method for studying microbial studies has been this operational taxonomic units, right? This works by identifying clusters of closely related sequences with a fixed identity cutoff. However, different regions or different sequences vary in their, for example, sequence identity percentage, and sometimes phylogenetically related taxa share a less predictable identity percentage. This is a limitation in this study, or in other words, this 97% that one uses to cluster these sequences into OTUs might not cover all of the sequences diversity, or might also include some of the closely related species. To address these limitations, in recent years, a denoising method has been introduced through several different popular algorithms as an alternative clustering method. This algorithm actually uses probabilistic method to predict actual sequencing errors, which are noise, before forming clusters. And hence, this clustering method is termed as amplicon sequence variance, and this is based on sequence probability rather than sequence identity. And this results in a fewer but self-consistent custom clusters that have been thoroughly validated to have more acceptable precision in many different publications. So, for example, based on mapping of these specific regions that are amplified from, let's say, 16S rRNA gene in the bacterial ribosome, these OTUs or ASVs can be assigned to belong to particular taxonomical classes of bacteria, and this will actually help us, for example, calculate the abundance of each one of those bacterial family or phyla in the community. And each community can be studied by comparing with the others between similar conditions or different conditions, or between different individuals, or between different state of these individuals, for example, healthy and obese, or generally, yeah, people with autoimmune diseases or not, for example. So these analyses will actually include in-depth understanding of data preparation, reference databases, and analytical approaches to, for example, to formulate hypotheses or to answer specific questions based on what we are actually understanding, or what we are actually identifying from these taxonomical classification. So importantly, this sequencing data, for example, will represent only one type of microbial omics, which are becoming many more, sorry about that. Yeah, this sequencing data will actually represent this metagenomics abundance classification, which will help us in, as we already discussed, in quantifying the abundance levels of different phyla or genus or species level classification of bacteria in the sample. So this is usually done by annotating or mapping these OTUs or ASV sequences to a reference database. And these reference database can actually include a ribosomal database project or repositories such as Green Genes or the Silva database. So these databases contain taxonomic information for the domains of bacteria, archaea, eukarya, based primarily on the phylogenetic information or phylogenes that are built from the small subunit rRNAs or 16S rRNA for the prokaryotes and 18S rRNA for the eukaryotes. So we can use this, for example, the Silva database to identify and to understand the taxonomical distribution of our OTUs. So what is the next step in this bioinformatics approach towards studying microbial abundance, right? So many questions about the role of microbial members or microbiome members and communities are now being answered using these next generation sequencing genomic data that we can generate. And this also can be done, or this in comparison or in complementary approaches such as mass spectrometry approaches for proteomics and metabolomics. Data generation can also be used to study this or related questions. So the next phase of this microbiome research actually lies in the ability to integrate these multi-omics data sets to combine not only the next generation genomic or metagenomic data, they can also integrate the mass spec data, or they can also integrate the structural data with phenotypical information about the overall changes in the community in their natural environment. So this is where the next few years or next few approaches of biomedical research in metagenomics and microbial population research is taking you to. So as we have discussed about the history of microbiology, the emergence of sequencing technologies, and how this data can be used to study, for example, abundance, microbial abundance in the samples. So let me show you, or let me share with you some of the additional resources that you can rely from the Learn OmicsLogic portal to study these topics in greater detail. And this will also include practical analysis tools that you can gain experience to in handling such data sets, right? Let's see. Let it work. Yeah, now it's so. We have courses on metagenomics, and we have example projects that take, for example, that include data from popular publications and explores their effects, or explores the data for their association with what is being studied, or with some novel associations that you can find from existing publications. For example, in Project 19 takes the Rett syndrome data, Rett syndrome microbial population that is estimated for children with Rett syndrome and for healthy individuals, and finds a dysbiosis and relates that dysbiosis generally with the dysbiosis that is characterized, symbiosis and dysbiosis that is characterized after subjecting the patients and population to variety of different antibacterial or antibiotic treatments. So that's an excellent project, for example, to integrate information from more than one source of publication. And we have several other projects, such as projects that focuses on human microbiome data, and a project that focuses on model system, like a mouse model system, so which we will shortly discuss. So let's talk a little bit about the approaches that you will be introduced to, and throughout this course you will be given access and directions to the server tBioInfo platform for bioinformatics processing and for analyzing these metagenomics data. This platform includes demo pipelines, and this also includes analysis approaches to run metagenomics bioinformatics pipeline. Different stages of this analysis are performed on different, what happened? Yeah, different stages of this analysis are performed on different sections of this multi-omics platform. So you can explore not only on this metagenomics area, but also the other ones when you are interested to do. So the interface of this server will enable you to quickly build logical graphs of analysis methods and sequence pipelines to understand each steps and their significance in this analysis. For interested people, the implemented pipeline can be accessed using the demo pipeline that I will show you how to do that. So let's quickly go through, for example, a demo pipeline of a project that we will definitely get our hands on in this program. So this project is a practical example of understanding the role of high-fat diet on obesity, right? So this is the project number 13 in the Learn OmicsLogic portal. So when you have, if you have the subscription, or when you get your subscription, you can get introduced to this project. This is an extremely interesting project where, oh wait, what happened? Yeah, so where the aim of the study is to test the hypothesis that the obesity associated microbiome undermines behavior even in the absence of obesity.